All right, good afternoon, everyone. Welcome to lecture 13 of BMS 300. Today we'll continue our discussion on the integumentary system. Before we get started, I have a couple of announcements. Announcement one is about the RPA activities. So previously I had mentioned, or the syllabus mentioned, that you have unlimited attempts of the RPA activities, and some of you said there were only 10 attempts, and so I looked into it, and apparently it was set to 10 attempts. And so I said, hey, you initially told unlimited, why is it 10? So they've increased it now to 20 attempts, right? So if you want to take the RPAs again and again, you can have up to 20 attempts. You only need maybe 4 or 5 attempts to see all of the questions in a bank, right? So I would say that take each attempt on a different day, and starting yesterday we have RPA 2 available as well. So if you just go back and forth between RPA 1 and 2, that will make sure that you're interleaving the activities. Alright, so, and also just another reminder, exam 1 will include integumentary system, skeletal system will be covered in exam 2. And so let's jump into today's material. So on Monday we talked about the question, how does the components, or how do the components of the integumentary system aid in its function? And this brought us to consider the 3 layers of the integumentary system. We talked about the epidermis in quite a bit of detail on Monday, and I told you what are the various cells in the epidermis, and now we move to the dermis, which is the lower layer. And in the dermis, we'll see that the associated tissues will include connective tissue. You will find nervous tissue and also muscular tissue. And the epithelial tissue is in the epidermis. So we see that all of the tissues which we considered will be found in the integumentary system. The dermis is also where you have appendages like hair follicles, sweat glands, sebaceous glands, and so on. So when you consider the epidermis, there are actually 2 layers. The first is called the papillary layer, and this is actually quite a thin layer. It's only composed of 20% of the dermis. And this papillary layer is made up of the areolar connective tissue. So the areolar connective tissue is what we spent, I believe, like around 20 minutes discussing it when we talked about various types of connective tissue. And this has collagen and elastic fibers, and also reticulate fibers. But this has a number of immune cells, and so it makes sense that if the epidermis were ever punctured, then the areolar tissue is right underneath with all of those immune cells. And what separates the papillary layer from the epidermis is the basement membrane between them. And as we go deeper, the majority of the dermis is made up of the dense irregular connective tissue. And so the dense irregular connective tissue has a number of collagen fibers which are arranged in a random fashion, and that's why the skin can be moved back and forth in multiple directions, and you have a strength against stretch in all of those directions. And there's also elastin fibers present, and that is why the skin can be stretched and it's going to recoil back to its shape. So now we will talk about what are the various appendages which can be seen in the dermis and in the various layers of the dermis. So in this cartoon, notice that the upper layer is the epidermis, and that's been removed, and the lower layer is the hypodermis, that's also removed, and we'll focus on the various components in the dermis shown here in blue. So notice that there are these projections called dermal papillae, and they will fit into these upper epidermal ridges, and that plays a role in the fingerprints which are unique to every individual. Notice also there are these capillary loops which are extending all the way to those dermal ridges, but it is from these capillary loops that nutrients can diffuse to the upper layers. And there will be these sensory receptors like Meissner's corpuscles, which we will talk about in the upcoming slides. And there are also free nerve endings, which is another type of sensory receptor which can detect pain. So these are some of the components in the dermis, but let's see how the fingerprints are formed. So the fingerprints, they are formed by the interlocking of the epidermal ridges and the dermal papillae. So both of those will lock in together, and the pattern in which they lock in is going to be quite unique from person to person. And so in the modern day society, we use this for personal identification to know if someone did a crime or not. But physiologically, these ridges, they play a role in increasing the surface area of the tips of the skin, and that can also help to increase friction when you're trying to hold onto something. And these will start forming in utero around 10 weeks of gestation, and it's unique from person to person because the way these ridges are formed really depends on the environment which is in utero for each individual. So while we are discussing about these the fingerprints, I think a related concept is the cleavage or tension lines, which are seen throughout the skin all over the body. Notice that these lines are formed because the collagen and elastic fibers, they are arranged in a parallel fashion, but notice that this parallel fashion is going to vary in different parts of the body. For example, in the upper part of the abdomen, they are horizontal, whereas if you come lower down, it's going to be vertical. So it really depends on which part of the body you're considering. And this is relevant especially when you consider surgeries. And if an incision is made parallel to these tension lines, it's going to heal without a scar. But if incision is made perpendicular to it, then a scar will form. And I have a habit of looking at my knee. I think every year or every semester I teach this to see if I still have the scar on my knee, which is perpendicular to these tension lines. And so I'm sure you may also have that experience. And if some scar is not disappearing, it may be because the injury was perpendicular to these tension lines. So with that, we'll move on to the various components of the dermis. And it looks like a lot of things are labeled here, but it's just the things which I talk about, which are labeled in red, is what I would hold you responsible for. So firstly, we see that there are blood vessels, and these will help to bring nutrients to the dermis, and from there it can diffuse to the upper layers. And here is a hair follicle shown, and that's the structure from which hair will grow out, and this is called as an appendage. At the base of the hair follicle, you have these glands called sebaceous glands, and these sebaceous glands will produce this oily secretion which lubricates the skin, but it also has an acidic nature which can have antibacterial properties. And we'll revisit sebaceous glands when we talk about acne when we come back tomorrow. I noticed that there are also sweat glands which can help to excrete substances like sodium and other wastes, and it can also help to cool the body temperature. And finally, notice that here is the erector pili muscle. This is connected to the hair follicles, and this is a type of smooth muscle. And whenever you have goosebumps, it's because these muscles will contract, and that's why the hair will stand on your skin. So notice that I've pointed out the muscular tissue. There's connective tissue on the upper layers. There's this epithelial tissue on the upper layers, and there's connective tissue throughout the dermis. And the last tissue which we need to consider is the nervous tissue. So there are a number of sensory receptors in the epidermis and the dermis, and we will talk about each of these and their unique functions. So the first type of sensory receptor is the Merkel cells, and notice that this is the only sensory receptor which will be found in the epidermis. The other sensory receptors will mainly be in the dermis. And the function of these Merkel cells is to detect sustained pressure or touching, because these receptors will adapt slowly to the stimulus, and so it takes a while for them to detect the touch. And the Meissner's corpuscles, which are found towards in the papillary layer of the dermis, they can detect light pressure. So this is another type of receptor that can detect light pressure. And as you go a little deeper, you find that there are these Ruffini corpuscles, which can detect warmth or the stretching of joints. And at the similar depth, there are also these Pacinian corpuscles, which can detect deep pressure. And keep in mind that the way I remember Pacinian corpuscles is that the Pacific Ocean is the deepest ocean, and the Pacinian corpuscles can detect deep pressure. And finally shown here are the root hair plexus. This is a group of nerve fibers which will innervate the ends of the hair follicles. And if something were to touch your hair and you detect it, it's because of these root hair plexus. The one type of sensory receptor not shown in this slide are free nerve endings, which can detect pain. So this here is an important slide. It's easy to ask a multiple choice question from this. All I do is mention the name of one of this, and you have to pick what is the function of that, or maybe mention a type of sensory input, and you have to pick which type of receptor can detect that. So this may seem like a lot of detail, but keep in mind that this will serve as the input to the brain. So when we move to the nervous system, we see that the input to the brain comes from these types of receptors. And then when we move on to the muscular system, we'll see how the brain can respond to the stimuli. For example, if a fly is sitting on your hair, or a mosquito touches the hair on your skin, you can swat it off. So the response to that input is from your muscular system. So with that, we move on to the deepest layer, and this is the hypodermis shown right here. As seen in this picture, you notice that there is a lot of adipose tissue, and one of the major functions of adipose tissue is to absorb shock. And this can also act as an insulator and reduce the amount of heat loss. And so if you don't have too much adipose tissue in the body, it can make you feel cold easily because you lose heat more rapidly. And this tissue can also serve as energy storage area, because the triglycerides can be stored in the adipose tissue. And this layer, it's technically not called the skin, because it is just the superficial fascia on which the epidermis and the dermis sit. So I'm sure you would have heard the term myofascial pain, and the question is, what is fascia? So fascia is just referring to the connective tissue which is holding the structures together, and the fascia and the muscles is going to hold the muscle tissue together. And some of this connective tissue has nerves in it, and that's why if you press on the connective tissue, you can activate the nerves, and that can trigger fascial pain. So coming down to what do we mean by the fascia as far as the hypodermis is concerned, we see that it's made up of two types of connective tissue. The first is is the adipose tissue, and the second is areolar connective tissue. So let me just quickly refresh you on the three layers which we discussed in the integumentary system. The epithelial cells are found in the epidermis. The dermis is made up of two types of connective tissue. What are they? Areolar and dense irregular connective tissue. And the hypodermis is also made up of two types of connective tissue, adipose tissue and the areolar connective tissue. So most of the fat stored in the body is going to be in this region, and once the hypodermis reserves are already filled up and there's fat stored there, then fat starts accumulating in other parts of the body, which can have pathological implications like fat in the liver, or in the heart, or in the gut area, and so on. So with that, we can move on to the functions of the integumentary system. So there are many functions of the integumentary system, and the first one is protection. And there are three ways in which the skin can protect us. Firstly, it can act as a chemical barrier because the sweat and the sebum, which is produced by the sebaceous glands, they can form an acid mantle, and that means it has a low pH, and that low pH can have antibacterial effects. So this is the first type of chemical barrier. The second type of chemical barrier is something we have discussed previously, and that is the melanin pigments which are in keratinocytes. Those pigments can act as a shield and protect the UV radiations from the sun from affecting the DNA in the nucleus of the keratinocytes. Secondly, we see that this plays a role as a physical or mechanical barrier, and we see that the epidermis has got multiple layers of skin, and that can protect us from abrasion. But also the keratin, which is produced by those keratinocytes, and the glycoproteins can prevent the loss of water. It makes the skin water resistant, and that means it's going to prevent both the entry of fluids into the body and also loss of fluids. And the importance of this, it will make sense when we talk about burns and other injuries to the skin. And finally, we see that it can serve as a biological barrier because you have immune cells in the integumentary system. So as far as the epidermis is concerned, we talked about dendritic cells. You move into the dermis, we have the areolar connective tissue where you have a number of immune cells. We talked about macrophages, mast cells, and then neutrophils, and so on. So there are a number of immune cells in the areolar connective tissue as well. And finally, defensins are also produced by keratinocytes, and this is a type of glycoprotein which has antimicrobial effects. So this is the first function of the integumentary system. Moving to the second function, we have the regulation of body temperature. So there are two ways in which we can sweat. The first one is called insensible perspiration, and this happens without you even detecting it. So when you're sitting in class or you're taking a nap, you're still perspiring and you're losing fluids from the body. And if you don't believe me, here's an experiment for you to try out tonight. Right? So before you go to sleep, weigh yourself, and then when you wake up in the morning, weigh yourself again. You would see a drop in your body weight, and that is because of insensible respiration. So let's say you're exercising intensely, then you can sense the perspiration, or if you're out on a hot day. Now, so this is the first way in which the body can regulate the temperature through the integumentary system. The second way in which it can do it is by directing the flow of blood either close to the surface of the skin or away from the surface of the skin. So let's say it's a very cold day, then the body is trying to retain the heat, and so we see that the blood is going to be circulated away from the surface of the skin. And how it does that is by causing the vessels which are close to the surface to constrict, and the vessels which are far from the surface to dilate, and blood will take the path of least resistance, and so the blood is flowing closer to your body core, and that's why temperature can be retained. On the flip side, let's say it's a very hot day and you're overheating and the body is trying to dissipate the heat, the blood vessels close to the skin surface will dilate and blood will flow there rather than the core, because the blood vessels away from the surface, they will constrict, and that's how blood flow is regulated. Any questions on this at all? how the body maintains the temperature. Okay. Third function is the cutaneous sensory reception. So there are various types of signals which go from the skin to the brain, and one of those is the sense of temperature, whether something is hot or cold. And an example of this is the Ruffini corpuscle, which can detect warmth. Also, different types of touch can also be detected by the brain through different types of sensory receptors. We see that Merkel cells can detect sustained touch, whereas Meissner's corpuscles can detect light pressure, and hair follicle receptors can detect if something touches the hair on your skin. And also, deep pressure can be detected by the Pacinian corpuscles, and pain sensation reaches the brain through free nerve endings. Alright, so this is how the integument can help with sensory reception. We also find that the integumentary system plays a role in metabolic functions. So we talked about this in lecture 4, what is metabolism. This is referring to all of the reactions which take place within your body for the sustenance of the body. So one of these reactions we consider is how vitamin D is going to be synthesized. And this is a really important concept because it fulfills the fourth learning outcome of the course, and that is trying to integrate what happens at the cellular level all the way to how different organ systems are being affected. And so when we talk about the synthesis of vitamin D, we will talk about the integumentary system, of course, that's what we're in. We'll talk about the impact on the digestive system, the urinary system, and the skeletal system. So skeletal, urinary, digestive, integumentary system, we'll see how all these come together for the synthesis and the utilization of vitamin D. So the food we eat has a form of vitamin D, that's vitamin D2. That is an inactive form, and only 20% of our required vitamin D comes from the diet. The remaining 80% comes from exposure to the UV rays of the sun. So when the UVB rays come in contact with dehydrocholesterol, this is a name I'll keep you responsible for, it's going to convert it into vitamin D3. And vitamin D3 will in turn go to the liver, where it can be converted into calcidiol. And calcidiol is also the inactive form, so that's sent to the kidneys, where it's converted into calcitriol, the active form of vitamin D. So the active form of vitamin D can in turn act on the small intestines, where it can cause the calcium to be absorbed from the digestive tract into the bloodstream. And where do you think calcium will be incorporated? In the bones, right? So that's how it all ties together. So we see the skeletal system is affected if the calcium isn't being absorbed, and we find that the role of the liver and the small intestine means the digestive system is involved. The conversion of calcidiol to calcitriol in the kidney means the urinary system is involved, and it all starts with the sunlight coming in contact with the skin. So you see how all these four systems are connected. Any questions at all on this slide? There's quite a bit of things I said. Yes?
What do you want us to know from this slide?
Great. So that was the last hour. No one asked me any questions, so I said, I know the question you have is what do you need to know in this slide. So thank you for asking that. So there's a lot of details in this, and I'll tell you what I will hold you responsible for. Firstly, I want you to know what are the organ systems involved in vitamin D synthesis and its use. So we know it, right? Integumentary system, digestive, urinary, and the skeletal system. So know what organ systems are being involved, and know what intermediate product is being converted there, right? So for example, know that dehydrocholesterol is converted in the skin to D3. I know that D3 is converted into calcidiol in the liver, and then calcidiol is converted to calcitriol in the kidneys, and that can in turn act on the small intestine and absorb calcium, which can be incorporated in the bones. So I don't need you to know the structures or these other alternate names, the enzymes involved in converting, which are involved in catalyzing the various reactions. That is beyond the scope of our discussion. All right, is this reasonable to know these details and how they all fit in? Yes?
Supplements of cholecalciferol, would that be sufficient for your bodily needs? Or just putting contact with the sun?
Yeah, so if you don't have sufficient sunlight, like for example in winter times in upstate New York, that's a real concern. So dietary supplements are necessary. But if you're living in Fort Collins and we have no shortage of exposure to sun, then your dietary need will be as much. But it also depends on what stage of life you're in. If you are immobilized and you can't go out, then you would need to take supplements. But most of the vitamin D needs can be accomplished by exposure to sunlight. And also, the interesting thing is that this direct sunlight, like if you are going for a drive and the glass can filter out those rays and from triggering the vitamin D synthesis. Maybe that's additional points. You only need to know what I discussed in this specific slide. Any other questions? Yes?
So let's say you go out and you get like excess sunlight.
Okay.
Is your body able to store like does your body store that if you have too much, or does it just excrete?
It is fat soluble, so after a certain point it can't store. But you would need it at least a couple of times in a week exposure, 15 minutes per day. Okay, so with that we'll move on to our discussion of the functions. Oh yes, one more question. Yes. Repeat that again. Oh, artificial exposure. Yeah, that's also possible, especially if you have had a friend who, or I heard of someone who had this lizard which needed sunlight exposure, and so they had an artificial lamp so that it could get those rays, UV rays for survival. So yes, for those who are immobilized, they may have UV lamps to make sure that their body produces sufficient vitamin D. Okay, so moving on to the other type of metabolic function, we see that there are some steroid hormones which can be produced by the skin, and if there's a dysregulation of their function, it can lead to autoimmune disorder. Right? So this is something I'm mentioning in passing, and I won't hold you responsible for details on this second point. But we also see that the skin can act as a blood reservoir. 5% of the total body's blood volume is held by the integumentary system. And the importance of this is if any part of your body has an increased metabolic demand, then that part of the body can be sent the blood. And the same principle also applies to how thermoregulation happens, by either making the blood flow close to the surface or away from the surface, and thereby either lose the excess heat or to conserve heat. So the same principle applies here as well. And finally, in terms of excretion, we see that the sweat which is produced can also excrete a number of nitrogenous wastes and salt as well. So these are the major functions of the integumentary system, which I hope you're all clear with. So here are some of the key questions which I believe you should be able to answer at the end of part 1 of the integumentary system. And so all of these questions can be answered using the summary slide shown here. Alright, so let me walk you through each of these questions. So the first question is, what are the various layers of the epidermis, and what are the unique types of cells seen in each layer? So the 3 layers are shown right here: epidermis, dermis, hypodermis. And in terms of the cells found in the epidermis, we have keratinocytes, immune cells, melanocytes. There are sensory receptors like Merkel cells. And in the dermis also you have a number of sensory receptors. And the dermis and hypodermis are made up of areolar connective tissue, and that is common to both. And dense irregular connective tissue is present in the reticular dermis, and hypodermis has got adipose tissue as well. And then the second question was, what are the two parts of the dermis and how do they differ in their composition? And the answer for that is dermis has areolar connective tissue and dense irregular connective tissue. The third question is, what are the different sensory receptors found in the epidermis, dermis, and elaborate on their unique functions, right? And so the answer for that question is, we talked about 6 types of receptors: Merkel cells, Meissner's corpuscles, laminated corpuscles, or Pacinian corpuscles, and then Ruffini corpuscles, the free nerve endings, and also the root hair plexus. So 6 of those each have a unique function. Go to that slide and you will know that answer. And finally, what were the various functions of the immune system? We went through role in protection, thermoregulation, sensing, reception, and hormone synthesis, excretion, and so on. Right. So, is that clear? How we can answer the key questions using this summary slide? Yes. All right. So, with that, here is the polar bear question, which you should be able to answer. The reticular dermis of the skin is an example of what? Oh, by the way, before we get into part 2 of the integumentary system, a trigger warning when we talk about some of the pathology and burn injuries. Some images may be disturbing, so just a heads up. Alright, so if you're squeamish, please be warned. And I think these images also sort of prepare you to know whether you should be in pre-med or pre-vet, because you will see similar things when you're in such professions. Okay, so most of you said dense irregular connective tissue, and that is the correct answer, right? So the reticular layer of the dermis is the 80% of it, and this is made up of dense irregular connective tissue. So let's have an overview of part 2 of the integumentary system. So we'll see how the integumentary system has a pretty good regenerative potential after an injury. So we'll see what are the various steps in the healing process. And then we'll talk about 2 types of injuries, namely burns and frostbite. And so, as of last lecture, I found that that's how far we are going to get today. And when we come back tomorrow, we will discuss about some of the age-related changes, which could be as you get older, or even acne, which happens when you're much younger, but it's an age-related change. And we'll discuss about warts, this condition, epidermolysis bullosa, and also skin cancer. Alright, so I believe tomorrow we'll be able to get through the material for the integumentary system. So when you think about the healing which takes place after an injury, it can happen in 2 ways. The first type of repair could be regeneration. The second is fibrosis. So regeneration is referring to when tissue is damaged and it is replaced with the same type of tissue. For example, let's say you have a cut on your skin and the epithelial cells have been damaged, and then those epithelial cells are replaced with more epithelial cells. That's an example of regeneration. Fibrosis, on the other hand, is when the damaged tissue, let's say the muscle was damaged and it is replaced by a scar tissue, that is known as fibrosis. And the concern with fibrosis is that the repaired tissue cannot perform the function which it formerly used to. For example, the muscle was damaged and you have fibrous connective tissue that cannot contract like how the muscle could contract. So now let's look at a few examples of how tissues are characterized based on their worst to their best regenerative potential. So in terms of the worst regeneration, we see that the skeletal and cardiac muscle are in this category, and even the nervous tissue will also fall in this category because it's going to be replaced by scar tissue. Now the reason why it's replaced by scar tissue is beyond the scope of our discussion, but if any of you are curious and interested, I'm happy to discuss that with you. In terms of cartilage having a poor regenerative capacity, keep in mind that this is avascular, so it doesn't have any blood vessels in it, and so that's why this doesn't have a good regenerative capacity. Now, having intermediate regenerative capacity will include smooth muscles and dense regular connective tissue. And what is an example of dense regular connective tissue? Where would you find this? It would connect muscles and bones. So, what is that called? Connection between muscles and bones. Tendons, right? Bone and bone ligaments. So, if you have, let's say, a sports injury due to tendon damage, if it's given sufficient time to heal, then it can recover. So they did a study on rats where they damaged the tendons and gave some rats 2 weeks time of rest, and the others did not have that rest. Those rats which had the time of rest were able to recover fully. So take home message for those of you who are who like to exercise, right? If you are adrenaline junkies who like the endorphin rush, if you get injured, I think it takes as much discipline to take rest and let your body heal as it takes to go out and exercise. All right. So with that, we'll see what has the best regenerative potential. And it's no surprise the bone tissue has the best regenerative potential. And the reason for that is not only the increased vasculature, but also the types of cells which make bone tissue. So we'll discuss that when we get to the skeletal system, maybe not this week, maybe next week, because I have a feeling we'll talk about the integument tomorrow as well. And then the epithelial tissue and loose areolar connective tissue, both of these are major components of the integumentary system. So it makes sense why if there is a damage to the skin, it's going to regenerate completely, because the major components of epithelial tissue and the loose areolar connective tissue have a high regenerative capacity. So now let's talk about the various steps involving a wound healing cascade. So there are 4 steps. We have hemostasis, inflammation, proliferation, and remodeling, and each of these will take place at a different point in time. Right? First one will take day 1. Inflammation can be for the next few days, and then proliferation for the next couple of weeks, and the last step can take many months. So as we go through these steps, keep in mind that I'm only going to give you very little detail on each of these steps. The one step where we will go into more detail is step 2, which is inflammation, and we'll discuss that in the immune system. But the rest, keep in mind that it can be very complex, and so for the scope of this course, I'm going to keep the details limited. So step 1 is hemostasis, and this is where you have the blood clot being formed, and the main goal is to prevent blood loss, right? So hemostasis. And how this is accomplished is by aggregation of platelets. And we'll see what platelets are. They're one of the components in the blood, and how they start aggregating is a very complicated process, right? So it can take up to 30 steps, and there are different players with numbers which don't follow the actual the numeric sequence. So if you take one of those advanced courses, you'll have the joy of memorizing it, and I will take my hat off to you, right? We won't get into that here. So another way to prevent blood loss is by vasoconstriction. So the blood vessels close to the injured region will start constricting so that blood is not flowing there, but it is redirected away from that region. And this blood clot is going to be formed, and that's what the scab is. And this scab will be made up of a protein known as fibrin. And this these steps will eventually initiate the step 2, which is inflammation. So in inflammation, we see that a number of white blood cells are going to be recruited to the affected site to prevent infection. Keep in mind that the injured region will have the areolar connective tissue, which has macrophages, and those macrophages can recruit other macrophages to that area by releasing certain chemical compounds. And once these white blood cells arrive, they can engage in phagocytosis to eat up those pathogens, but it is also going to release growth factors, which will lead to step 3, which is proliferation. And so as these growth factors are released, we find that in the proliferation step, there will be fibroblasts entering into the affected region, and it starts producing collagen. So when did we talk about fibroblasts previously? We talked about what tissue? Connective tissue, right? So fibroblasts are what help make the surrounding matrix, and so one of the proteins produced by them is collagen, and so that starts producing the matrix around the affected region. And then the epithelial cells will also start proliferating and dividing, and blood vessels will form if that has been affected as well. And following proliferation, we see that the last step of remodeling takes place, where the collagen fibers which were produced in the previous step are rearranged, the blood vessels will be strengthened, and the scab tissue will be replaced by scar tissue. And we see that the epithelial cells, the lower layer, they're going to eventually push forward and they will get rid of the scab tissue, and you have the scar tissue remaining there. Yes, what's your question? Could you speak up? I can't hear you. For the slides? Yes.
So for inflammation,
Yes.
I think you had a copy of it
For the information. Oh.
Yeah, so like it's the same thing. We basically the same design.
Okay. Is there a mistake of facing? Thank you for pointing that out. So I was told that there may be a mistake in the handout I posted. Is that right? Yes. It's okay. I believe you. And I'll just go back and make sure that the text matches correctly. Thank you for pointing that out, and I'm sorry for that. So moving on to the couple of type of injuries, let's talk about burns and frostbites. So a burn is said to occur when tissue damage happens in response to exposure to heat or to electricity, radiation, or certain chemicals. And one of the first things which happens is the proteins in that specific region, they will be denatured. And what we mean by the term denaturing is that the proteins will unfold and their function will be affected. We talked about protein denaturation. If you take a fertilized egg and you put it on a frying pan, it gets denatured, it can no longer become a live chicken. And in addition to these proteins being affected, that can also impact the integrity of the cell, and that can cause cell death. So when a burn happens, there are two outcomes. One is an immediate outcome, the other one is a delayed outcome. The immediate threat is that there can be a loss of fluids, and if you have blood loss, that's going to have an impact on your circulatory system, because without the blood being pumped around, it can lead to circulatory shock. It also can affect the kidneys, because the blood pressure has to be maintained in order for filtration to continue. And if you have a loss in blood volume, that drops blood pressure and the kidneys will start failing. And this is something we will discuss in greater detail when we talk about the renal system and the cardiovascular system. And after the initial crisis, we see that the next long-term threat is exposure to infections, right? And so that is something we'll discuss in detail in the immune system. So notice that the types of burns can be classified based on which layer is affected, and so we'll talk about each of these briefly. So first degree burn is shown here, where you have only the epidermis affected, and it's shown here in red. And in this case, you have redness, swelling, mild pain, and causes of this could include like a sunburn or touching a stove. Any stimuli which has a very brief exposure that can lead to the first degree burn. A second degree burn is where you have not only the epidermis but also the dermis being affected, and this can result in greater pain, more redness, but also notice that there can be blistering because the blood vessels, the lower layers, their integrity is affected, and so the fluid can start accumulating from those vessels. And this could be caused by a moderate exposure to a moderate stimuli, and it could be like chemical burns which cause this. And finally, in the third degree burn, we see not only the epidermis, but the dermis is fully affected all the way down to the hypodermis. And this can result in dry, leathery skin, which is charred, but also this can be painless because all of the sensory neurons are in the upper layers, and so if those are destroyed, then you don't feel pain in that region. But keep in mind that the surrounding region of this third degree burn may have second degree burns, and so that can still be very painful. And so this could include fire victims, electric burns, and so on. So let's spend like maybe a minute looking at a few pictures and see if you can identify what kind of burn it is. It's not for testing you on the exam, but I just want to make sure that you got the gist of what I was saying these last few minutes. So what do you think? This is 1, 2, or 3? First, second, or third? Okay, most of you said second degree, and it's because of the blisters. Very good. And what about this one? First degree, because it's just a sunburn, right? And the last one, what about this? Third degree burn, right? Because the skin is charred. And what about this, folks? Second degree burn. You will notice that the surrounding regions seem to have like a first degree burn, but you always classify based on the latest degree of impact. So moving on to frostbites, we see that this is something which we will experience in a few months, whether we like it or not, the cold weather. And this is an article from earlier this year on how there was an increased occurrence of frostbites. And one of the reasons this happens is not because people aren't taking enough precautions, but many times they just don't have the resources to protect themselves from the cold. So frostbite is also known as freezing cold injury, and this happens when you have exposure to 32 degrees or temperatures below that. And I know many of us have different levels of cold tolerance, but depending on the level of exposure, it can lead to this type of injury. And similar to the burns, we see that frostbites can also be staged depending on which specific layer is affected. Right? So if there is a first degree frostbite, it's called a frost nip. There's skin numbness, pain, tingling, but keep in mind that there is no long-term impact. For a second degree frostbite, you could have these milky blisters, erythema, and edema. And edema is where you have fluid accumulation because the lower blood vessels have been affected, and erythema is where you just have reddening of the skin. The third degree frostbite is where you have digital edema, where you have swelling along the edges of your digits, and there's also hemorrhagic blisters because the lower the levels you go to, the hypodermis can also cause blood to be released into those blisters. And the fourth degree is where even the bones are affected and you have necrosis, where the cells die due to lack of oxygen. And in this case, all the digits and the thumb also had to be amputated, right? And many times when people experience fourth degree frostbites, it's because they've experienced near-death experiences and they're just thankful to be alive, right? I watched a video of where a hiker was stuck in the cold, his partner had died, but he was just thankful to be alive, even though they had a condition like this. So with that, we'll come to age-related changes when we come back tomorrow.